Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast, part of the Finding Genius Foundation. I have Jonathan R. Woodward. He's a professor, part of environmental sciences at University of Tokyo. And we're going to talk about magnets interacting with human cells, which may shed light on how animals migrate, but I'll let Jonathan describe that. So welcome, Jonathan. Thanks for coming.
Hi there. Nice to meet you. Thanks for having me.
Yeah. Tell me about your research.
Okay. Well, probably the reason that I'm talking to you is that some of our recent experiments have made a step forward in what we're looking at. In our recent work, what we found is that we can take living cells. So the cells we actually use are a very famous kind of cell used by researchers, which are called HeLa cells. They're actually human cancer cells. And we can grow these cells. And all cells have various molecules and components in them that if you shine light on them, will actually light up slightly. They'll glow. And all cells do this. This is a well-known phenomenon study.
Really?
Yeah. So this is one way you can see this. So you can take any kind, just some regular cells. If you shine the light on them, they will slightly glow. The thing to realize that this is very tiny. You really need a very special microscope to see that. You can't, you wouldn't see it just by look. But what we did was we were able to demonstrate that the amount that these cells glow, the amount that they fluoresce, changes when you put the cells in a microscope. So what we actually do is we develop this special kind of microscope, and we watch as we shine a blue light on the cells. They emit green light. This glow is green, and it comes from a series of molecules known as flavins. And those molecules get excited, and they emit this green glow. But what's interesting is that then how much they glow depends on what else they're getting up to. So they're busy doing other things, and we can actually use the amount that they glow to work out what other chemistry. And it turns out that by applying this magnetic field, we're observing some other chemistry that they're doing. They're doing magnetic field dependent chemistry in the cells. So this is a really interesting. This is kind of the first time that anyone has been able to see a regular cell responding in real time. So what we do is we switch the magnetic field on and off and on and off, and we can see the fluorescence get dimmer and brighter, dimmer.
Wait, how are you inducing changes in the magnetic field? It's with light or with changing magnetic field?
No, we apply the light continuously. We shine light on them, and then what we do is as these cells are glowing, we switch a field on and off. So we have a very special electromagnet that allows us to make a magnetic field at the sample at the center of our microscope, and we can apply that a field in any direction, and we can change its strength, and we can do different things with it. So what we do is we turn that magnetic field on and off, and we can actually see almost with your eyes on the camera, but certainly in the data very clearly, that as you apply this field and switch it on and off, that the brightness of the light coming from the cells.
Well, why would you have to hit the cells with light at all? Why not try varying the magnetic field to see what's going on?
So the light is what you're using to measure the chemistry. It's very difficult to. So what we're actually trying to do is watch chemical reactions as they're happening inside the cell, which is an extremely difficult to do. You need some way to watch the progress of the reaction. Now, it turns out that in physics, in chemistry generally, fluorescence of molecules, this property is a very, very common way to monitor what molecules are up to. So that's what we're doing. We're using this natural glow that exists in the cell as a means of tracking the cell's chemistry. And then we're observing directly the changes in the chemistry by applying.
Well, why not use fluorescent tagging of the cells? Would that kill them? Would that work just as well or no?
But I mean, so the point there is that one of the things I think that's important about this is you've not touched the cell in any way. So the moment you put a fluorescent tag in and then say, we can see change with the magnetic field, you'll always be subject to the criticism, well, you've changed the cell by putting this. So the idea here is that...
So this is a less invasive way.
Yeah, so this is brilliant. You're using the cell's own natural properties to track its own reaction. You don't need to modify it in any way. You don't need to mess with it. Of course, there are methods. And actually, one of the other things that we're working on in our lab in a completely separate project is trying to make magnetically sensitive, very bright fluorescent tags for molecules that tell you about what's going on, not just by their fluorescence, but also magnetic field response. So that's another thing that we work on.
Before you start modulating the magnetic field, the light that's hitting the cells, what kind of chemistry is that causing to happen just without any modulation of magnetic field?
The point here is that when we shine the light on the cells, it's initiating these photochemical reactions in these molecules. So the chemistry that's going on is the same, and then what the magnetic field allows us to do is make modulations. So we then observe those in the chemistry as modulations in the light. So what's really critical here, and why we think this work is important, is that when you shine light on cells, then the molecules in those cells are undergoing photochemical reactions that proceed through short-lived species known as radicals. And those radical pairs are at the heart of why the light becomes field sensitive. And those are interesting, those radical pairs, because they are what are proposed as probably the most likely suspected mechanism by which some animals may be able to sense.
So what kind of chemistry do you see happening? This is in human cells you're looking at, or what model are you using?
The cells that we use are called HeLa cells. They're originally human-derived cells. They're from a cancer patient years ago, and they're what's called an immortal cell lines, and they're a very good model cell for modeling normal cell behavior that researchers use. So the beauty of these cells is you can freeze some, and then you can grow new cells from them at will.
All right. So again, what kind of chemistry do you see going on? Is stuff happening in the mitochondria, or what's different about the cells when you modulate the magnetic field?
Okay, so it's probably more important to talk about the chemistry itself. So, what we do in my lab and what we've done for many, many years is study. We're not, I'm not a biologist originally. I'm a physical chemist, and what we study is these kind of species called radicals. And radical pairs are fascinating species because their chemistry depends on a very, very fundamental property of matter called electron spin. So, and that's why there's an awful lot of interest in this, because basically the reason that a magnetic field can affect them is all to do with the spins of two chemical relatives. So I can talk a little bit about that.
Again, the chemistry in biological terms, what kind of chemistry seems to be affected, and what is its function in the cells? Before we go into the specifics of it.
Entirely, we have no idea. We don't yet know if this has physiological consequences or not. That's all future work to be done. So what we know at the moment is that basically all cells contain molecules in them called flavins, and they have flavins in different forms. Okay, so the flavins are a very important molecule in biology. They're used by many proteins as what we call a cofactor. And for example, flavins are used in plants, in enzymes called photolyases, for example, to repair DNA when it gets damaged by light. So, all the details of all the flavins in cells is still, very much an open piece of research. People are still understanding more and more about this. What we can say so far from experiments is that the flavins that are naturally present in the cell, when they're exposed to light, they get photoexcited by the light, and then they generate these species called. So that's what we understand. What we don't know, it's a very important point that you made. What we don't know yet is whether that has any consequences to the cell. So does that get turned into a response in any important biological process? That we don't know.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on support us. We have 3 levels of membership, from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click support us today. Now back to the show. So what's the first steps now in your experimentation? You're modulating the magnetic field. One question I wanted to ask you beforehand is, how does the frequency of light affect the cellular response with or without the magnetic field? Have you tested that?
Yes, of course. So, I mean, that's a critical part of what we do. So basically, this is the standard thing in chemistry. This is spectroscopy. So what we carefully control is the wavelength or the color of the light that we're putting in. And we look very carefully at the wavelengths of light that are emitted, the glow. And so what happens is if you put different colors of light into your cells, you're going to excite different molecules. And we choose our light carefully to be, what the light we use is 450 nanometers, which is blue light. That light is about where our flavin molecules get most excited. So we can target the flavin molecules by using that light. And then if we look at the light that we get sent back from the cells, then that is shows a characteristic dependence on the wavelength. We measure it at lots of wavelengths, and we can determine that it has exactly the light characteristics that we expect from these molecule flavins. So part of the work we do here is to demonstrate for sure that it's the flavins that are giving this magnetism.
Well, if you know that it's flavins, what biochemically, what do flavins typically do in cells?
Well, like I said, they're in many things and they have many roles. So exactly what, and we're hitting the whole cell here and looking at what's going on. So there are different, and I mean, one of the important things is that, some cells have different flavin containing molecules and there are some that are in common. All right. So in the quest, for example, to understand how, for example, birds navigate, the proteins that are proposed in that are called cryptochromes, and the heart of cryptochromes are flavin. And as I said, the flavins are involved in a wide range of different chemical procedures. They're in various different proteins. So in our experiments at the moment, we don't have an ability to dissect which flavins or exactly what they're doing that are giving the response. And so that of course is something that we need to start looking at in the future. So one of our important next steps is to try and identify, exactly where the flavins are that we're seeing. So I mean, inside which proteins or inside which kind of environments in the cell, and then untangle the chemistry.
Well, on a slide, can you have cells that are illuminated and adjacent ones that are not, but yet they're still subject to the same pulse of magnetic field, and see if there's any difference?
Well, so again, this is a real-time experiment. So you can in this experiment, you only see what's happening to the cells while the light is on, right? While they're fluorescent. So, and our microscope is designed specifically so that we irradiate only one region of one cell, the region of interest that we're in, during the experiment. So we deliberately, focus our light onto a region of the cell of interest. So, but I mean, yeah, of course, one thing we can do now is, of course, we can look at the effect of, we can try and look for downstream effects on the cells by applying them to magnetic fields or not. One of the problems that there always is when you're concerned with magnetic effects in biology is that when you try and do kind of statistical analysis of cells and things like that, you're getting down to the limit of the effects that you see. I've been working, actually, when I was a PhD student, I was trying to look for many years ago, look for magnetic responses of chemistry in cells then. And we were using those kinds of approaches, where we, for example, look for DNA damage in cell by measuring lots of cells afterwards and seeing how much damage and whether the field effect. It's very difficult. It's very difficult to be sure. Some data looks like there's a real effect, and other data looks like there isn't. And it's very hard to pull out from statistics. So the beauty of this experiment is you don't have to worry about that. This is much more a chemistry experiment than it is a kind of a biological statistical assay. You see the effect directly in real time. You can see it happening.
So you're not illuminating the entire cell, you're just illuminating a subsection of the cell?
That's right.
Well, why not do the whole cell? What would be different about that, you think?
If you like this podcast, Please click the link in the description to subscribe and review us on iTunes.
Well, I mean, in the case of the HeLa cells, they have a very particular shape, and they have a very big nucleus, and we don't see any fluorescence. So we're actually you see the other parts of the cell around it, and we just then, because of the shape of the cells, we kind of optimize the size of the spot that we look at to get as much of that in a single image where we're sure it's the same region. And then we can, we measure many cells and we look in, but this is the region of interest. Yeah, there's just basically a big black hole in this.
So this region is what, just in the cytoplasm, or where does it appear to be localized?
So we see, I mean, it's very clear from our images that, it's that we see it, for example, in the mitochondria. So in our images, for example, you can see the mitochondria moving, and that they're alive, this thing, and they're quite brightly. So we haven't done an analysis yet of exactly where the fluorescence. Again, that's future work. But our key suggestion from it is that we want to look at potential flavin containing species. That's
so. Again, what have you noticed so far with the changing magnetic field? What chemistry appears to be turned up or turned down or happening?
Again, this comes back to this is the species known as radicals. So it's a very complicated process to explain. Pretty difficult to. But the idea is when you excite your flavin molecule, then what happens is a nearby molecule. In our study, we still don't know which molecule the partner will be yet, and that's one of the key to find out. In cryptochrome, for example, it's a tryptophan residue in the amino acid tryptophan. And what happens is this molecule donates an electron. It tosses an electron to the excited. And what that means is that the flavin molecule now has one more electron to begin with, and the other molecule has one less electron. And normally, most molecules in nature have an even number of electrons. They're all paired up in the orbital of the molecule. And when this electron toss happens, it means it means both these species now have an odd number. So one of them's got one more than normal, and one of them's got one less. And what that means is they have these unpaired electrons. And unpaired electrons in chemistry are important because they expose the properties of the electron. So that's what you're seeing in here. And this pair of radicals. What you actually see is radicals themselves are then dangerous. All right, radicals are very reactive species. So if these radicals go off, they can go, cause damage elsewhere. But what is largely happening is once the molecule has tossed the electron, after a short time, the flavin tosses it back again. But its ability to toss it back is what's influenced the field. So what the magnetic field does is it controls the total spin state of these two radicals in this kind of quantum mechanical process, and that's what determines how quickly the.
So where in nature would people be affected by this, or I guess other animals? Like, the Earth has a magnetic field, I guess incredibly weak, but is that enough? Like, have you tried an experiment where you just changed the magnetism, around a set of cells so that it just counteracted, let's say, the Earth's magnetic field, and to see if the chemistry changed there? Make it appear as if there is no directional magnetic field of the Earth. Would that cause any biological response?
So that's a very good question, and so this is a little complicated because when scientists working in the area of radical pairs do this, okay, so what we know from radical pairs is that as you change the magnetic field, they don't respond linearly. All right, they have their main response. In most cases, their strongest response is in the strength of the kind of strength of fields that we looked at. So that's what you look at. It's in the order of tens of millitesla, a few millitesla, tens of. That's a lot stronger than the Earth's magnet. Okay, but we also know that radical pairs, and many of them, when the conditions are right, they can also show a completely opposite effect at much weaker magnets. This is what's called the low field effect. And it's that low field effect that has led to radical pairs being a key candidate for explaining, for example, animal magnetoresistance. So there have been studies, there have been chemical studies that have shown that you can make a kind of designer molecule that will generate radical pairs and that will show magnetic responses of the kind you described, right? So that if you switch on and off the Earth's magnetic field, the molecule, you can observe a response. But so far, that has only ever been done in a kind of designer molecule system. There has never been any study that's done that in a natural molecular system. Okay, so then if you move to real molecules, then scientists have studied the cryptochromes, which is this protein from birds and from other animals that's been implicated in the sort of magnetic field response to the Earth's. And they can see these radical pairs formed in there, and they can see the magnetic effect in there. But there has been no one so far has been able to observe effects, just in the molecules themselves in a test tube at these very, very weak fields. And that's an ongoing experimental and theoretical challenge. There's a huge debate in the field about what's the case. So there's something missing in the story. I don't want to kind of conflate our story too much with that story, because while we're addressing flavins and radical pairs and cells, the bird navigation story has a much more specific twist. It's looking at the cryptochromes specifically and looking for the response you described specifically.
So you may find effects, but then that obviously, as usual, it may not necessarily translate to biological systems because there may be, hidden interactions, but at least you're seeing that there's effects.
Yeah, so we've seen
at a high magnetic field level relative to the Earth's magnetic field, right?
That's exactly it. So the argument goes like this. Radical pairs predict, let's say, two kinds of effect, one at very weak fields and one at stronger fields. And we can see radical pairs responding to stronger fields in cells now. So that's the step. So, of course, one of the obvious next steps is, okay, can you see a weaker field? And the reason we've not done that yet is that you have to be very, very careful cancelling out the Earth's magnetic field. It's non-trivial to do that and be sure you're doing it correctly. So our current magnet system is, excellent for applying slightly stronger fields, but I don't think it can give us the level of certainty that we've completely cancelled the Earth's field. So we will build a new set of magnetic field generating equipment to work at these because you have to, of course, think in 3 dimensions. You've got to cancel the field along exactly the same axis, which depends on where you are on the Earth and which way the Earth's field is pointing. So you've got to work very hard to get rid of the Earth, and we will do that.
Well, I guess there's 2 ways to do it. I just realized one is the only fields that you apply are weak ones to try to counteract the Earth's magnetic field. But another way to do it is to counteract the Earth's magnetic field at the weak level, but then apply strong level magnetic fields on top of that.
Yes,
and I guess try to wipe out that background signal, right?
Yeah, that's exactly what you do. So basically, yeah, your job. I mean, you're doing it all with the same coils, but once you've built a system where you can convincingly control the field in 3 dimensions and completely counteract the Earth's magnetic field, you can then superimpose another field in any direction of whatever strength you're generating. So yeah, that's exactly what you would do. So you can switch the Earth's field on and off, and then you can apply smaller fields in the Earth or bigger fields.
But what's the end goal of the research that you're doing? What do you want to figure out?
Well, so there are a number of things. Obviously, this, as I said, this has been an ongoing concern for me. I'm a chemist. I've been studying, and I do in many other projects, study radical pairs in chemical systems and biological systems. And for this particular research, where we now, I mean, this is a question I've wondered about for 30 years. Can we get this magnetic field response? You mentioned it already. Does it have a consequence to the cells? All right. Is there a downstream biological effect that's processed in some way? Are there any health implications then of these kind of effects? But basically, we work in a kind of blue sky science way. We are just trying to understand what's happening. It's absolutely remarkable that there's a quantum mechanical mechanism that would allow chemistry taking place in cells to respond to something as weak as magnetic field. So that in itself is fascinating. And now what we want to do is understand that in all its forms. So we want to know exactly how it's happening, exactly what the molecules are, exactly what the mechanisms and the rates of the processes are. We want to understand it in detail. And then, of course, once you understand something properly, you can then start to look at the implications and also the application as well. So, this could be, I don't know whether it is or not, it could have implications for health, both positive and negative implications for health and magnetic fields. It can have...
Well, there have been many studies. Many studies on electric fields on the human body.
Right.
I mean, we're bathed in nowadays modern times are bathed in all kinds of EM and magnetic fields and everything. So,
Absolutely.
I mean, in your experimentation, do you also do you incorporate that? Because you'd want to, any biological system nowadays, unfortunately, at least us, maybe not as much animals, but they're in a different background environment from, 100 plus years ago.
Yeah, that's true. So that's, yeah.
You're into experimentation as well.
Yeah, so I mean, with all these things, you're limited. You can't switch off the outside world, right? So you're always going to be doing experiments in the environment in which we live. But what we can do is isolate these effects. I mean, most effects of electric fields in the environment are essentially screened out by cells just because of the way the dielectric properties of the materials in there. So it's usually not an issue. But that's why people are very interested in whether it's the magnetic field from electromagnetic fields in our environment that can cause an effect, because that's not screened now by anything else in the cell. So it can enter right inside. And now we see that it does, in fact. So, but you're absolutely right. This is a very interesting question, but there are good reasons in most cases to not expect a lot of the things in the environment to affect us.
Offline, I asked you about quantum biology and, your thoughts on it. So if you don't mind, can we restate that? You started to point out the difference between quantum biology from a biologist's point of view versus a chemist's point of view versus a physicist's point of view. So if you don't mind, just let me know your thoughts about quantum biology in general.
Sure. So, I mean, I think quantum biology itself is this term that's kind of sprung up in relatively recent years, and it's a sort of general term that tries to bring together non-trivial quantum effects in biological systems. And I think that it has lots of different reactions by different people. And I think what I was saying to you before is it really depends on your training, your background, and your expectations. So it's, in my experience, the physicists I know, when they study quantum physics, they're very much focused on the sort of pure mathematical behavior and very, very small objects and doing very, very precise quantum measurement type effects. And there's a sort of sense in which seeing any of those amazing quantum effects in big, hot, wet systems is something that's surprising. And then at the other end, that you've got biologists who, of course, much more familiar with people with things working in a normal classical sense. And most of our original understanding in biology has been along those lines. But it's always the case. I think if you talk to many chemists, in particular physical chemists, in their training, they study quantum mechanics basically as the first thing, because it's the rules that underwrite all of chemistry. The first thing you need to do to understand chemistry is to know what an atom is and what a molecule is, what a chemical bond is, and all of those things are fundamentally quantum mechanical. So, and there are many aspects in chemistry. If you take something like isotope effects on chemical reaction rates that we've known for a very long time, those are pure quantum phenomena having really clear macro scale effects. You can measure them. They're very useful. You can do useful things with them. Can you give a brief idea of, what do you mean the isotope effect? People that don't understand. Okay, so for example, in chemistry, there's a very famous phenomenon known as the kinetic isotope effect. And what that means is if you do the same chemical reaction with two different chemicals, one of which you've substituted one of the isotopes in. So let's say you might have replaced a hydrogen atom somewhere with a deuterium atom. Then remarkably, those, depending on the chemical reaction itself, those reactions can take place at quite different rates. One could be one could be faster than the other. And the origin of those effects is purely in quantum mechanics. It's either in a change in the 0 point vibrational energies, or there are other sort of equilibrium effects you can get as well. But this is well known, for decades and decades. And now one of the aspects, for example, of quantum biology is seeing those kinetic isotope effects happening in enzymes and in real biological systems. So that's one of the examples. But I think from a chemist perspective, we're kind of used to that. And the idea, quantum physics looks at the microscale, but what chemistry does is it looks at how the microscale affects the macroscale. That's really one way of looking at how chemistry works. And so I think a lot of chemists are a little bit underwhelmed by the term quantum biology because there doesn't seem anything particularly remarkable in it. They might have expected there to be observable quantum effects in biology.
So, all right. So tell me about the experimentation you're doing. Let's recap it in this new light. Let's go over it and I ask you questions.
Sure. So, okay. Well, let me start with the mechanism that underpins kind of all the research in my lab. So, and certainly this work. What we're interested in, what we've been studying for many, many years in my lab is these species which are known as spin correlated radical pairs. Okay. And these species are, well studied, well categorized, and we see many, many, many different examples of their behavior in many, many different ways and in many, many different systems. But the basic idea, and I can't remember, I think we discussed this somewhat last time, but the basic idea is that most molecules are filled up with electrons. They have a whole series of electrons inside them with different energies. And for the most part, most molecules, all those electrons are actually paired up. So most molecules have an even number of electrons. And I'll explain what I mean by paired up in a moment. But what you can do is, it's possible to do a reaction, for example, between two molecules, where you get one molecule to give an electron to the other. And the result of that is now that you've got an odd number of electrons on each molecule. And it turns out that very often that means is that you've got one unpaired electron in each molecule.
In chemistry, I guess the sharing of an electron is more of a covalent bond, and then an ionic bond is, are you describing what's called an ionic bond, or is this something different?
Well, okay, yeah, so I'm not discussing, but you're absolutely right. I mean, in the normal case, let's imagine it exactly like that. Let's say you've got a covalent bond, all right? Let's talk about this slightly differently. Okay, let's say you've got a bond between two atoms. Let's just take hydrogen as a simple example. It's not a realistic one in this case, but let's take the hydrogen molecule, so H2. There's a covalent bond between the two hydrogen atoms, right? Yeah. And what we could do is we can break that bond. We could break it in one of two ways. We could break it so that both the electrons go on one hydrogen and the other one's left with none. And that would leave you with an H plus ion and an H minus ion. Or you could break the bond symmetrically. So each hydrogen gets one electron, and you'd be left with two hydrogen atoms. And those hydrogen atoms then have an unpaired electron, and they're what we call free radicals. So, and what you would have done is generate them in a pair. Does that make sense?
Well, okay. So, yeah, through chemistry, like you said, you can create these different scenarios. What's common in nature with hydrogen, for instance?
Well, I mean, normally we're talking about deliberately breaking chemical bonds here. Okay, so normally, both happen quite commonly. And for example, in biology, enzymes very often use this what we call homolytic, where you break bonds evenly and create radicals. They often use radical routes to transforming molecules inside enzymes. So radicals are common, and ions are common. But what's really important here is that the idea that you create these two radicals together in a pair, because just simply from counting, you go odd plus odd equals even, and you start from even. So you generate a pair of radicals. And this is where you get into the interesting quantum mechanical situation, because each electron, I should say a little bit about electrons for the audience. So electrons are species, they're fundamental particles with a number of properties. And some of those are very familiar to us. So we know that they have mass. We know how much the mass of an electron is. And they have charge. Electrons are negatively charged. And those are familiar. But electrons also have another fundamental property, just like mass and charge, called spin. Yeah, spin causes a problem because lots of people don't really understand spin properly. Spin's kind of complicated. You have to really put some effort in to understand spin. But spin is a purely quantum mechanical property. There's nothing equivalent to spin in classical mechanics. So it's a purely quantum mechanical property. And the name spin is both a blessing and a curse.
Can you talk about spin? I mean, what is it? I believe it creates...
Yeah, I'll do my best to give you a brief discussion about it, but, it's a little complicated. Okay, so let's electron spin. Okay, so the name itself comes because we like to try and understand things, particularly quantum mechanical things, by analogy with things that we do understand. So the best way to start thinking about spin is to think about the Earth going around the sun. All right. And as the Earth goes around the sun, It's moving, and it has a property in physics, a classical property known as angular momentum. All right, so this is, well, when we learn in high school, when you push a truck along of mass Vm and you push it at velocity v, it has a linear momentum mv. When something moves in a circle or something similar in some kind of orbit, it has this property called angular momentum. Okay, and so we call that angular momentum orbital angular momentum because the Earth is orbiting the sun. But it's really then important to realize that the Earth actually has two kinds of angular momentum. It has this orbital one because it's going around the sun, but it also has another kind of angular momentum, which is called spin angular momentum, because as we know, the Earth rotates on its own axis, and that motion gives it another angular momentum known as, which we would call spin angular momentum. And so this word was borrowed because electrons in atoms, at some level, you can kind of think of them a bit like the Earth going around the sun. So you've got electrons moving around central nucleus, and they actually have angular momentum called orbital angular momentum, which describes how they move around. But they have this other property, which looks very like angular momentum and behaves in pretty much the same way, but it's not something we can control. So if you could stop the Earth spinning, you would lose that spin angular momentum. But with electrons, the spin is built in. You can't make it go away. It's a fundamental thing like their mass or charge. So all electrons have this built-in thing, which we call spin. It's sort of a kind of angular momentum, and an electron can have it two different states. All right. So it can have two different spin states, and it can only exist in essentially when we measure it in one of those spin states.
Okay. So now going to your experiment, what are some of the particulars of it?
Okay, so there's this thing then, the radical pair mechanism, and I'll explain it to you in a moment. But the radical pair mechanism was discovered, so it was first proposed in the sort of 1960s, and then in the 1970s, people predicted that it should be able to lead to magnetic effects on chemical reactions, and indeed, those were discovered. And so what it does is it relies on, it's a very specific mechanism, and it relies on these spin properties of the two electrons, and I'll explain it in a moment. But what's been very interesting is that in the 1970s, this model was proposed as a model by which animal navigation might work, by which animals may be able to sense magnetic field. And that was dismissed and ignored for a long, long time. But became much more popular in about 2000, and now people have really started to believe it. So let me go on and address how this works. So if we go back to the situation we discussed a moment ago, we've got a hydrogen molecule, say. When we break it in half, we have two electrons. We have one electron on each hydrogen. Okay, and there's a very, very fundamental law in chemistry called the Pauli principle, and what that means is that inside the molecule, inside the hydrogen molecule, in the covalent bond, the only way to have a covalent bond is if the two electrons in there have opposite spins. So I'm going to refer to those spin states as up and down, just for simplicity. So if you've got one of the electrons spin up and the other one spin down, they're paired together, and that's fine. You have a chemical bond. And the key thing is when you break the chemical bond, the two radicals remember this, all right? They know what the spin state of the other one is. So this is similar in some ways to some of the kind of quantum communication and teleportation experiments you hear about in physics.
Quick question. At the time of the bond, does an entanglement happen, a quantum entanglement?
So, yeah, I've kind of avoided talking about it because in many ways, some of these radical pairs, depending on what state they're in, we can write them, they are entangled. But really, the entanglement is kind of coincidental. It's not the entanglement that really is at the heart of the radical pair mechanism, which is why I've not kind of deliberately gone down that line. But yes, there are four states basically that the radical pair can be in, and two of them are entangled states. But I think it's probably not helpful to get too much into that. But in principle, you can do like quantum teleportation experiments with radical pairs, and that's been done a year or so ago. So the idea is that you've got these two, and they remember. And basically, in our experiments, what we do is we use the blue light in our experiment, and we excite this molecule called a flavin. And when we excite the molecule, a nearby other molecule, usually something like a tryptophan, throws an electron, and the flavin catches it. So now you've got this radical pair in the same way. So you've got a flavin with one unpaired electron, and you've got the other molecule. And where the magnetic field effect comes in is that what happens is the electron can only go back if these electrons remain paired. So if the spin state on the two species, if the two electrons have opposite spin states. But what we know is that over time, due to the interaction of those electron spins with nuclear spins in the molecule, so this gets really complicated, that drives changes. So, for example, one of the electron spins may flip over. When it does, the two radicals now don't have paired spins, and it turns out to be impossible because of this thing called the Pauli principle for the electron to go back. And so in our experiments, what we do is we toss this electron over, and then we put a magnetic field on, and that changes the ease with which the electron comes back. And when the electron comes back, the flavin can get excited again, and it might fluoresce, it might give out light. But if it's stuck in this other radical pair state with the two spins with the electron not able to go back, then basically it can't go back to its fluorescing state. And so you see the effects of this process going on in the chemistry of these radical pairs, and it's written into the fluorescence. And that's what we're seeing in this experiment.
It's a little bit difficult to understand, but is there any clinical significance, or how do you think this is going to translate into something that can be harnessed and used?
So, I mean, it's a question to which I don't have a great answer at the moment. I mean, certainly, as I've said, the great promise is that this mechanism is the mechanism that's underpinning, for example, the magnetic field sensing ability of animals. So understanding that's key. But I mean, certainly from our perspective as well, we are one of the projects currently in my lab is to design very, very, very efficient molecules that are incredibly easy to detect with fluorescence, so that we can detect them down at the single molecule level. They actually have strong magnetic field responses due to these radical pair effects. And then what we can try to do, for example, is build probes that we can put inside real biological systems. And we can, as well as having the light response, we can use the magnetic response to report on the environment. So that's one sort of practical application. But being able to build kind of biological systems that are, effective sensors of magnetic fields, because it seems the animals can have this huge acuity to detect tiny fields. So there are technological applications, I guess, in things like navigation for aircraft and stuff like that as well.
Well, so if you look at the magnitude of the Earth's magnetic field, how does that compare to the sensitivity of these animals? Is it well within the limits where they could detect small fluctuations in the Earth's magnetic field?
I mean, that's the remarkable thing about the biology. One, straight off, the Earth's magnetic field is very, very small. It's something like 30 to 70 microtesla, depending on where you are on the Earth. It's really, really, really tiny. And animals, all the evidence from biology is that you can really detect these tiny, tiny fields, and in some cases you can map. Some animals are able to sort of make magnetic maps that can respond to, small changes in the angles and the sizes of these fields. Now, in our understanding in the radical pair mechanism, we're still struggling to explain how that can be done with such acuity. And that's a really ongoing problem in this field that many people are working to try and solve, both experimentally and theoretically. But if we could build chemical systems that had such acute responses to the magnetic field, those would be very, very interesting and very, very useful.
So what kind of animals appear to be navigating using the Earth's magnetic field? And, physically,
well, there's actually
what structures in their body do it?
Okay, well, I mean, again, this is a very much a contemporary area. I mean, probably the best studied are migratory birds. In particular, the European robin has been studied extensively, and it's well understood in that sense. The other one that's been very well studied, because it's a kind of good scientific host, is the fruit fly, the Drosophila fruit fly. Fruit flies show magnetic field sensor orientation effects. Some of the other really amazing ones are turtles, which basically seem to use magnetic fields. They lay their eggs and then travel in a huge circle for about a year, and then are able to swim back to exactly the same spot where they laid their eggs. And actually, if there are shifts in the Earth's field, you can sometimes see the turtles go back to the shifted location when they get back a year later. But there are many. There's a very interesting paper out this month on looking at the effects in butterflies. There have been huge range of studies that have been done. When I was working in this field when I was younger, I remember the paper that came out because it was when basically Google Earth and Google Maps were first launched and became popular. Scientists had a new data tool, and some scientists had basically analyzed the statistics of the direction of cows in fields. And they had suggested that statistically cows aligned themselves along the magnetic field lines. So there's a huge amount of research that goes on. As you know, I'm not a biologist, so I don't read every single biology paper, and I'm not up to date on everything, but there are a wide range. I mean, right down to bacteria. There are what are called magnetotactic bacteria, and they work in a different way. They actually grow these tiny magnets inside themselves, and they swim around in a magnetic field. You can find amazing videos on YouTube of them doing it.
Wow, that's crazy. So you're hoping to make, like you said, molecules that are very sensitive to, fluorescent analysis when they're, I guess, injected or swallowed by a person for what medical conditions, let's say.
Yeah, so we want to use that to be able to, I mean, there are lots, I think we mentioned it briefly before, but there, the idea of having fluorescent tags in biology is a very, very common technique. What we're trying to do is develop some kind of fluorescent tags that give you more information because they show a magnetic field response which reports on the environment of the radical pairs. So that's one thing we're working on, and we've got some very promising results on. Yeah. So of course there are many other things too.
What kind of biological mechanisms do you think you can, look into if you're able to successfully do this?
One of the nice things that what these probes will do in a nice way, if we can get them to work in the way we'd like, is that they can be, for example, we can decouple the light response, which reports on certain things, from the field effects. And what the field response will give us largely is very good indication of the environment. So, for example, how viscous is it? You would expect to see very different responses from these things if they were in a membrane, for example, or if they're in something like cytosol. You could use the same probe throughout a cell, for example, and you could differentiate regions based not just on the light response, but on the magnetic response as well. So, I mean, this is kind of right at the beginning. So we will wait and see exactly what applications they can have. But in chemistry, we can use radical pairs in very useful ways to give us very detailed information on processes that are happening nearby. So they can report, for example, on other magnetic species nearby. They can report on, heavy metal ions. There are lots of things that radical pairs can report on in a kind of interesting way.
Okay. What's ahead for you in the next year or so in terms of experimentation to get to like a clinical application?
Well, I mean, again, sort of biophysicists or chemists. I mean, for me, it's not something like, clinical applications is not something I've ever thought about. And maybe that's something that I have to really start thinking about now. So our real goal at the moment, certainly with the work that we started talking about here with the cell autofluorescence, is to really understand the details of that. So we know some of the story now. We know that it's flavins. We don't know what the other radical is. We don't know which particular molecules may be dominating. Is it particular proteins? We need to know whether, as I said before, there's any implications of those effects to the physiology of the cell. So is this just a coincidental effect that we've noticed, or are there consequences in other actually, health consequences for the cell and so forth? So there's a lot of work to do on that. And of course, we're doing other work where we're sort of combining those studies with using cryptochromes in cells to try and investigate the animal navigation stuff. And then, we're working on, I have different groups in my lab working on different projects. So we've got this, as I said, these sensor molecules that we're trying to build. And we're also doing a lot of work on these magnetic field sensitive polymers at the moment that we've developed. So we've not published any of that yet, but we've got some very exciting stuff on some new kind of polymerization reactions. So.
Okay. Well, very good. Jonathan, what's the best way for people to find out more about your work? Where can they go?
Yeah, the best place is my lab's website, which I will share with you. You can share notes. It's address is it's opes.tokyo.u-tokyo.ac.jp, and then it's /spinchem, SPINCHEM, So if you go there, you can find the group's website, and there's information on the news and that we're doing and stuff on there. So that's probably the easiest link to get to what we're doing.
And when you said C, do you mean the letter C or the word SEA?
Sorry, it's OPES dot letter C dot U hyphen TOKYU .AC.JP SpinChem.
All right, very good. Okay. Well, Jonathan, thanks for coming on the podcast again. I appreciate it.
No, thanks for having me. It's been a pleasure talking.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.